Okay, the second part of this chapter is cell parts, and we're going to kind of go over this somewhat fast because most of this should be on review for you. So I'm going to start, I guess I'll start in the center here. Why not, right? So in the center here we have, this is the nuclear membrane, right? Whoops, probably better share my screen, right? There, the nuclear membrane. And the nuclear membrane, notice that it has these open spaces, so we call these nuclear pores because we have to be able to let things out of it. And inside here, so I'm going to probably put some color in here. So okay, so this stringy would be DNA, and this is in the form of chromatin, okay, when it's stringy. So I want to make a note off to the side. So when DNA is in the stringy form, it is chromatin. When it's all balled up into chromosomes like this, we call it chromosomes. That's when we get ready for cellular division, okay? So that's important to note. This large piece of green on the inside here, this is the nucleolus. This is made of rRNA, okay, ribosomal, okay, RNA, okay, rRNA, and it makes ribosomes. Okay, its job is to make ribosomes. That's it's making the ribosomes, and the ribosomes are coming out of the nucleus. And then, of course, those are the sites of protein synthesis. Okay, so pretty important piece. It is not making any of the rest of the like tRNA or anything like that, but that ribosomal RNA is what makes up the ribosomes. That's what is in here. So you might see a dot within the dot under a microscope. That's really what it is. So there's our last organic that we were talking about was DNA, and that's located within the nucleus. So the next part, of course, then I want to share with you. This is one of the canal systems, right? And put some ribosomes out here, friends. And they're just scattered. I mean, imagine that the ribosomes are just scattered throughout the entire cell. And remember, those are just factories everywhere. If they are located on this piece, the one I'm drawing right here, then of course we would call this, you know this is the endoplasmic reticulum, or ER. There is rough and smooth ER. They both have a slightly different job. Okay, the rough ER has, of course, ribosomes on it. And it's a transport. This is the streets of the city. Okay, you can have smooth ER. I'm going to color it the same color, only because I want you to understand that it is ER, but it's a little bit different as far as its job. The smooth ER doesn't connect necessarily to the outside. The smooth ER contains enzymes to break down lipids or to make lipids, contains enzymes to make lipids. So it like synthesizes lipids. Now, why would you want lipids? Well, it's used for the cell membrane, right? And then don't forget about our friend, the ribosomes. Now, what good are ribosomes? Ribosomes are your making factories. These are your protein factories. Super important. Okay, protein factories. So remember in DNA, RNA, and this was from chapter 2, the big aha moment is that the DNA codes for what amino acids. Where are the amino acids put together? The amino acids are put together at the ribosomes. So this has the code or the blueprint. Ribosomes read and put the amino acids in order. So your DNA tells how your proteins are going to be made. If there's a mistake in the code or in the plans, then there's going to be a mistake at the factory. If you're putting all that together, which is kind of crazy. Okay, let's see what else we're needing. Let's put in, of course, our friend mitochondria. And I'm going to color them red just because they look like a bean. And of course, the mitochondria, you can't say mitochondria without saying powerhouse of the cell. This is, of course, where cellular respiration takes place. Okay, I do have a little aside that we're going to do. So I'm going to say, hey, flip your paper over real quick on mitochondria, because there's an important theory that's about mitochondria. So I want to talk about it because mitochondria are kind of weird. So we're going to take a break and move over to this side real quick. And I want to talk about something called the endosymbiotic theory. And the endosymbiotic theory is like a theory I've heard like in every bio class because it has to do a little bit with evolution. So something to keep in mind is that mitochondria has a similar membrane to the cell membrane. Okay, so as far as a phospholipid bilayer, it also has its own DNA. So it has mitochondrial DNA. How weird is that? It also can go through cellular respiration. So time out for a second. It has its own membrane. It has its own DNA. It can do cellular respiration. Sounds like it could live on its own. Well, that's the thought. The thought is that it was living on its own to have been and unfortunately it got engulfed by a larger creature and of course replicated in that creature and then was passed on. And that's how mitochondria became part and did not exist outside of as its own cell. What's really interesting is I've heard of some different shows that they've talked about how when you get half of your genetic material from your mom, half your genetic material from your dad, but your cellular material comes from your mom. So your dad's gives the genetic material, but your mom gives like her mitochondria. So when you look back, you can actually trace your mitochondrial DNA back to your mom's lineage, which is totally wild, which is actually pretty cool. So it's supposed to be the thought or the theory is that it used to be on its own, but we're pretty lucky that we have it because mitochondria is pretty important. Okay, let's keep going. So let's see, what other things do we have of the cell? Well, we have, let's do Golgi bodies. So kind of make them look like stacked pancakes. Or this is the Golgi apparatus or Golgi bodies. I'm going to make mine yellow in color. Think of the Golgi bodies as the UPS store. Okay, I love the UPS store. I love Amazon. I can take things back and they will package it back up for me and send it back. So this stores and packages. Okay, so it's like the UPS store. It's pretty awesome. And then we have other storage kind of units. We have vacuoles. Kind of three things that are very similar, actually very similar in shape. So I'm going to color code them, but here's the thing that I don't want you to worry about. So we're going to talk about like our project that we're going to work on cell models. We're not going to really worry about what it looks like. We're going to worry about what it does. Okay. So we'll call this first circle. This is going to be a vacuole or the word vesicle. And this is a storage. It can be a storage of waste. It can be a storage of food items. It can be a packaging, putting like the trash bag out. It can be all of those things, okay? Then we have two very similar sacks like this as well. We're gonna call this one is a lysosome. Now that kind of sounds like Lysol, doesn't it? They contain digestive enzymes. And these digestive enzymes will break the cell down and also break other vesicles down if need be. Okay? So this could do something we call cell suicide. So if a cell if a cell is breaking down or has to break down something or break itself down, the lysosome can actually self-digest. Another place, lysosomes. So again, breaking those down. Something else that is very similar to this would be a peroxisome. Now, a peroxisome sounds like peroxide, as in hydrogen peroxide. So this actually breaks down hydrogen peroxide. Now, you might say hydrogen peroxide, well, that's good for the cell, right? Well, remember hydrogen peroxide, we use that for treating cuts, right? But you shouldn't just use it willy-nilly. Remember that hydrogen peroxide, when you use it, it not only kills the bad stuff that might be in a cut, but it also kills good stuff. So be very, very careful when we're using that piece. Hydrogen peroxide is a byproduct of cellular metabolism, and that can actually kill the cell. So peroxisomes actually contain enzymes to break down hydrogen peroxide because it's toxic to the cell. It's kind of wild and crazy, isn't it? Throughout the whole cell, we of course have to color. I guess I'm left with brown. The jelly-like fluid. Now, keep in mind. So we're going to talk about that. The jelly-like fluid. We're not gonna create cell models where you come in with jelly and we're going to put it throughout the cell. That's not what we're going to do. But this out here is the cytoplasm. This is the site of most chemical reactions. That's reactions, okay? So what's happening when you stop thinking about what's going on with all the ribosomes and stuff out in the cell, that's really where all the action is taking place, is out there. Okay? The other piece that we need to talk about is the rest of how we're keeping all of these things organized in the cell. One last part that I want to note in here, I'm going to put a star just because when you see the pictures, that's kind of what these look like. This would be the centrioles because we'll mention those when we actually get into mitosis. These are responsible for cell division. These are the lassos of cell division. Okay, and these centrioles are actually made up of the cytoskeleton that we're going to talk about right now. So I left this space here. So now we're going to talk about the The cytoskeleton, oh my goodness, I've got things falling everywhere. So I'm going to put a little block here. Okay, so the cytoskeleton of the cell, so a skeleton is just like in the human body, it's a place of attachment. We have two pieces. So think about the cytoskeleton as like small bones and big bones, kind of. So we are made up of microtubules and microfilaments. Okay, within the cell. So one of my favorite things, hold on a sec. One of my favorite things to look at in your textbook is this picture of what the cytoskeleton looks like. Because when we all made a cell model, we all put it in a Ziploc bag, okay? And it was all jelly and it just was willy-nilly all over the place. That is so not what the cell looks like. Look at, this is what the inside of a cell looks like. Isn't that crazy? So there are all kinds of, it's like scaffolding inside, okay? It kind of reminds me of like your house, your studs, your beams in your house. That's exactly what's going on inside the cell. Now, friends, we're not going to draw that on here, but we're going to make note of what these are. So we have, these are the thin ones, and these are the thick ones, okay? Microfilaments are made of very small. They're made of two proteins, actin and myosin, okay? Where microtubules has tubulin is the protein. They're actually made of proteins, okay? And these microfilaments right here, these are responsible for muscle contractions, okay? So muscle cells are filled with actin and myosin, okay? So this is the thin stuff. These would be the small studs, where the microtubules would be your bigger beams, okay? So these are going to be small and thin. These are going to be much larger. This tubulin, these thick ones, these are what make up the centrioles that I just mentioned, as well as flagella and cilia. So when we make our cell models, you'll have to decide whether you want to be cilia, okay? This would be cilia, or whether you have a flagella. Now, friends, that's two L's, by the way. So what cells of the body have a flagella? Oh, sorry, ladies, it's not us. It would be sperm. What cells of the body have cilia? Well, that's easy. This is your respiratory tract. Actually has cilia. Okay, so here's our big thing that I want you to think about. So this is all of the cell parts. These should have all been very, very familiar to you because I know that you covered these in biology. So when I want you to think about making a cell model over the weekend, okay, in the next couple days, we want to make a model based on function, okay? A model based on function. So what we're going to do is you're going to do like a little mini video for me, okay? And you're going to show me the parts of the cell that you've decided, and you're going to tell me why you picked those because of the function. So I am not expecting like the plastic bag, and I'm not expecting, you know, something, so, oh, this looks like a tube, so this needs to be my microtubule. That's not what I'm looking for. So what I'm looking for is, can you build something? So maybe you want to think big. Maybe you want to go outside and be like, you know what, my yard is a cell because around my yard is a fence, and it's semi-permeable because it lets some things in and some things it keeps out. And think like on a bigger scale. okay? You should not be creating anything, okay? You can go around your house and come up with items that you think, you know, like you got Lysol, like here's hand sanitizer. I could say this is my Lysol. This kills off germs, or a bottle of hydrogen peroxide, and this represents the peroxisomes. Or hey, this is my fridge. I want you to start putting common items to these cell parts because the function is so vitally important. Okay? So that's what we're going to kind of look at. So while you're going through this, really think about what things do I have around the house that can be a cell model. On a normal year, we'd get together and we'd all bring them in, but we've got to do this kind of on our own, and it's really important for you to make that connection. Okay, friends? Thanks.